So welcome to the second talk of this morning session, and we are pleased to have Nir Gov from Weizmann Institute, and he's going to talk about random cell migration. Hey.
Thank you very much for the invitation, and thank you for listening. It's a big change from the previous talk, which was very mathematical and very interesting. This is about trying to take a physics-based modeling for how cells migrate. So it's mixing of physics with biology. And the part which is most relevant to this workshop regarding anomalous diffusion will only be sort of hinted at the end. So I hope it'll still be interesting, and there's open questions and possibilities for looking at this behavior in terms of anomalous diffusion, which we haven't done yet. So what will I be speaking about? I will be speaking about phenomena such as the one that you observe over there, where a cell has been placed on a linear track, meaning that there is a linear track here which you don't see with your eyes, where there is adhesive substrate on which the cell can adhere and then start scrolling. And everywhere on the sides, above and below this linear track, is coated with non-adhesive substance so that the cell cannot go either up or down, but stays in this linear track. And you saw that while the cell is moving persistently to the right, its back was making sort of stick-slip behavior, meaning it was remaining at the back until it gave up and then started moving. You can see that running again. Other examples, of course, include the same kind of geometry, but with multiple cells. So now you have cells that are themselves making some persistent motion, sometimes changing direction, like this cell has changed direction, divided, and interact with each other. I will get to this kind of geometry, which we are now very much interested in, which you can see by the way the cells are moving, that's actually an hexagonal network that is being painted here on the substrate instead of just one linear track as was here before. And now the cells have to deal with these junctions while they are moving around. So now they are actually moving in the two-dimensional space, which is spanned by this hexagonal network. And you can ask questions about how are they moving around. Now, what's the motivation for that? Why are we doing all these? Well, I'm not doing the experiments, I'm doing the theory, but why are people looking at that? And the motivation is that things like that actually do happen in the body. So in this case, you see some blood vessels, and the green objects are glioblastoma, brain cancer cells, a terrible disease, unfortunately. And they move around, they spread around in the brain by moving along these very thin blood vessels. They use them as sort of highways for spreading. So there is motivation, and you can even see one of them actually reaching a junction here, a T-junction, and having to decide whether to go right or left. The theoretical work that we did was actually motivated by this nice experiment which was called the cell race, where the organizers of the race asked people to send them their fastest cells for a competition, putting them all on these 1D lines, and then they looked for interesting correlations between the cell speed and the persistence of the path. And they found that there was a correlation, and that started us thinking about how to model it. So I will start by giving you a very brief, but I hope still comprehensible, description of the basic modeling that we did for how a one-dimensional cell is actually moving along such a line to model a behavior like that. And then I'll transition to the more interesting recent work on extending that into networks. So allowing this one-dimensional object to actually bifurcate and have many arms. So again, this is the object we want to model. And the work that, the theoretical work that I will describe to you was done together with Rafael and Jonathan, which was a student at the time in my group. And it appears in these publications. Okay, so the basic biology that you just need to know, and it's very brief what I will tell you, is that cells are of this sort that I will be talking about are propelling themselves and moving themselves forward by utilizing this machinery, which is called the actin cytoskeleton. So there's hundreds, probably thousands of proteins in the cell, and hundreds of them are involved in what is called the cytoskeleton, which is this network of biopolymers, of these elongated polymers that form from proteins binding themselves together to form these linear polymers. The same as the polymers that you're wearing, only that your synthetic polymers that you're wearing are made of subunits that are covalently bonded to each other, so they're very, very strong. That's why plastics are so useful. These ones are weakly bonded to each other, so the cell is continuously forming these polymers, but is able then to take them apart and reuse these basic building blocks to build the same polymers and the cytoskeleton somewhere else. So it's constantly remodeling itself. And the beautiful thing is that if you make these polymers polymerize close to the membrane, close to this thin membrane that is bounding the cell, and you can make this polymerization happen only, for example, in the front, in an asymmetric way, so only in the front of the cell and not in the back, you can actually utilize this directed polymerization to push the cell forward. And that's what is observed here. So in the front of the cell, there is a continuous polymerization and depolymerization in the back of these polymers, and that continuous internal flow of this matter from the front to the back is propelling the cell forward. Now, there is, of course, an energetic cost, so you have to pay with fuel, and the fuel of the cell is ATP. So continuously, the cell, while polymerizing and depolymerizing these polymers, is utilizing ATP, converting into ADP. Of course, the idea is that the cell is always in good health, has lots of sugar in the solution or whatever it needs in order to produce fresh ATP out of that ADP, so it can continuously be alive and sustain that motion. So you're not running out of fuel. The idea is that these cells are healthy and alive, and you can continuously do this internal flow, which in the rest frame of the cell actually looks like it's emanating in the front and moving all the way to the back. Yes.
On the linear track, they're always moving in one direction. Do they backtrack?
Yes. Okay. So the cells, when they're moving in one direction in those linear tracks, they do change direction. I will talk about that a little bit. But they do have persistence. So it's not on very short time scales, depending on the cell. Some cells don't move at all. Not all cells know how to move very efficiently. Okay. So the idea of the model is captured in this cartoon. So the idea is that if you break the symmetry of the cell by some mechanism, you can sustain it by the following mechanism. If you polymerize those filaments in the front and they flow towards the back within the cell frame, you can think of some molecule, which we don't specify yet what it is, that can bind and unbind from these moving conveyor belt of these polymers. So the idea is that these polymers polymerize in the front and depolymerize in the back. We don't describe that in detail, but there is some given velocity of this conveyor belt of filaments from the front to the back. And what it does is that it affects these molecules, because these molecules, when they are free in the solution, they diffuse, and when they are bound, they are advected with the flow. So there is a diffusion advection description of these molecules. If you assume, that this system can reach steady state, then you can actually solve for the profile of these molecules, and the profile is a simple exponential such that they accumulate in the back and they are depleted from the front. So the simplifying assumption that we will assume is that in all that I will show you is that the changes in the velocity of this conveyor belt is the slow process, and the fast process is the diffusion of this internal molecule, so that at all times we can use the steady state distribution of these molecules, and the only dynamic variable is just the velocity of this actin flow, which will be the slow variable that affects everything. Okay, so we're reducing the dimensionality of the problem. So what we find is, for example, is that if we now link back this distribution, this concentration of this molecule to the flow in a simple way, for example, you can assume that this molecule is an inhibitor of the actin polymerization, so that if the concentration of this molecule is high, it inhibits actin polymerization. So if it's pushed to the back, actin will not polymerize in the back, and because it's depleted from the front, actin will polymerize in the front. So you can write an equation like that, which gives you the local actin polymerization speed, either in the front or in the back, and how it depends on the local concentration of this molecule. And then you say that the net flow is simply the difference between the flow from the front minus the flow from the back. And if you write that equation, you can have an implicit equation for V, because V is the result of this right-hand term, but the right-hand term has V in it because all these distributions are dependent on the velocity through these exponentials. If you solve that numerically, you find that now the system can have a spontaneous bifurcation, where, for example, if your control parameter is the length of your system, if the system is too short, you cannot build enough of a gradient over such a short distance to self-sustain this speed. And even if you perturb the system to have some gradient, it will go back to constant concentration and zero overall velocity. But above a certain critical length, you can now have a bifurcation, and the system can self-sustain a cell moving. So we have a simple model for how a cell can transition itself from not moving to moving in one dimension. I will pick it up now and tell you how we now extended that simple model to allow this object to change its length. So now the length is not anymore a control parameter that is fixed, but it's actually calculated from the balance of forces that are applied on each end of this one-dimensional object. The assumption is very simple, that the object is like a spring, so it has some elasticity. There is some drag, which is applied only at the tips. We can apply it also along the whole length, but that was just a simplified assumption. To capture this behavior that you saw, that the back is not simply dragging smoothly along when the cell is moving, let's say, to the right, but sometimes it can stay behind and then do this stick-slip behavior. We can capture that by having the drag not as just a constant, but as arising from some adhesion molecules that can bind and unbind. And the assumption is that they are slip bonds, which means that the more you stretch those bonds, the higher is the off rate, the higher is the rate at which they will unbind. And that very quickly can get you a behavior which is like an instability, in the sense that if you think about it, if you have a certain number of these bonds which are holding the back and one of them rips, then the entire force which all of them were holding and sharing between them is now shared by a smaller number. And since a smaller number is now feeling the same force that was before, each one is stretched even more. So there's a higher probability of one of them unbinding even higher. Once that one unbinds and so on and so forth, you can get a catastrophic avalanche, and that's this stick-slip events. So now we have a system that we don't control its length, it's elongating by itself, and you can see that, for example, you can have a system where, as function of this parameter which gives you the strength of the flow, you can have a system which only elongates so far, and that means that it has not bifurcated because the two ends polymerize actin at the same activity. But beyond a certain point, again, it's elongated enough to break the symmetry, and now one end, which is the back, polymerizes very little, and the front polymerizes very strongly, so there is a net flow, and the system begins to move. That is illustrated here. So, for example, you can start with a very small cell. It begins to elongate equally symmetric on both sides, but it cannot reach this critical length LC, so it just stays not moving. If you cross that critical length, you can get into this stick-slip regime where you see nicely that the front is moving almost smoothly and the back is undergoing these catastrophic events of detachment and reattachment, detachment, reattachment. What you see in this case is that after each stick slip event, you never go back to being so short that you are losing polarity. Remember, below this critical length, the cell loses its polarity and doesn't know what is front and back, because the solution is that it should have the same polymerization from both sides, so it doesn't have any intrinsic polarization. So that happens here. You can actually have, after each slip event, that the cell shrinks back to such a small length that it actually loses polarity for a short while. If that short time that it loses polarity is too short, it actually remembers the polarity it had previously, and it continues maintaining this persistence. It all depends on what is the typical time scale by which these actin flows actually relax back to the steady state solution. The steady state solution tells you that you should have no polarity here below this critical length, but because you had polarity before and the velocity, these internal flows, have some intrinsic time for reorganizing and readjusting, it depends on this time, and of course it depends on noise. So again, if the noise is strong enough, during that short time, you can reshuffle the velocities such that you come out of this event without knowing where you were going before, and now it's just a 50-50 chance of changing direction, just like you asked before. So using this model, we could explain many features that were observed in one dimension for just a simple cell moving along a track. And what was nice is that there was then this experiment done by Amrinder in Virginia Tech, where instead of 1D lines, he was actually making cells move on a tightrope. So these are fibers, super thin fibers on which the cells are moving. What is interesting is that in the wild type, the cells actually exhibited stick slip behavior, and when he knocked out some component which is very important for recruiting actin to the cell front or the cell back, but to the edges of the cell, he saw that the cells were moving more slowly, but actually not showing any more stick slip, but actually moving very smoothly. And that's exactly what we expect in our model if you tune down this parameter, which is the parameter of how strongly the actin is polymerizing. You move from stick slip to smooth but slower motion, so exactly like the observations. Furthermore, you also you can also look at the persistence time. How persistent is the cell to continue along this direction or to change direction? And intuitively, you think that the stick slip motion could be the less persistent, because after each slip event, you become very short and maybe you lose your polarity. But it turns out that under these conditions, the model predicted that actually the stick slip is still more persistent and faster than the smooth motion of the knockout, and that was actually also observed in the experiments. So you get some taste that with this 1D model, we can capture a variety of migration patterns of cells along 1D, including random diffusion, such as captured by this persistence time. Okay, now let's move towards the networks. So before we talk about a highly bifurcated cell, the simplest bifurcation that you can think of a 1D cell is just to split it into two arms in a junction. And that's what you see here in this symmetric 120-degree Y junction that was constructed on this hexagonal network. So yeah, many faces, because many people contributed. And the idea was to focus it not so much at what happens when many cells interact. That's too complicated for us at the moment, but just to focus on what a single cell is doing in the junction. So basically, we took the same model that I showed you before, and we extended it, we generalized it. So now the idea is that the flows that is pushing this chemical from one side to the other is starting here. There is a flow, and then it splits into two. So the assumption that we have in the model is that each contributes a flow that is contributing 100% while it is within this arm, but it's split exactly into two when going into the two arms. And when this network is symmetric, that's a very logical and assumption which conserves all the flows. So under this assumption, we can go back and do the same model that we did before, and we found a very interesting behavior. Let's first maybe look here. First of all, we found that if the cells are not polymerizing, acting strongly enough, that's the y-axis, or they cannot elongate enough because they are very stiff, and that's the x-axis, then they are short and they don't have enough activity to elongate, and they just become trapped on the junctions. And that's interesting because these cells are in a condition where on a 1D line, they can polarize and move. But when you're on a junction, the junction acts as a trapping site. So that was already something that if you stop to think about it, it makes sense, but was not intuitive from the beginning, because I have a cell that moves, it reaches a junction, it has more options where to move, but it gets trapped. Why is that? And the reason is that it can get trapped because now, instead of just the front and the back competing to who will be the winner, you have another one which is competing. So you have more competition between all of them, and if the conditions are such that the polymerization is not strong enough to break this symmetric solution, you get trapped. Of course, if the polymerization is strong enough, then you escape and you go through the junction smoothly. I'll show you some examples. And then when the polymerization is even stronger, you can actually get transiently trapped in this very funny state where the two new arms that you grow into the junction as you come into the junction are just very, very long and are of equal length. The back is completely shrunk to zero, and there's still this indecision because both of them are super strongly polymerizing actin, and eventually there is something that breaks the symmetry. So that's the general behavior that we found, but if you look more closely at what happens, it's actually even more interesting. And just to remind you, there's no noise in this system. You can see that all the velocities are completely smooth. There's no, I'll show you examples of what noise does in a second. So if you let a cell spread on such a junction, if again you're below the critical activity, you just spread equally in all three directions and you don't reach the critical overall length, and that's it. You're just not moving anywhere. If you're above this critical activity, you see that the length crossed this critical overall length. The black is the total length, and you have here three graphs with three colors for the three arms that are growing. You have here three arms that are growing, and you see that at some point one arm is losing and two arms continue to grow. At some point the second competitor also begins to lose, and only one overall competitor is winning, and that's the direction, let's say it was this one, in which this cell will now go on and move and leave this junction. But what's interesting is that you see that once this one started shrinking, the green actually started growing. So there's actually an interesting oscillation on the way to making a decision. And you can even see it more strongly with plotting the local flows, the local actin polymerization activity at the tips. At first, it's the same in all three of them. Then the loser, which was the green, you see it's getting shorter here, has a drop in this polymerization speed, and the red and the blue are beginning to be the new winners. You can see that they are continuing to grow. At some point, you see, when the red was losing, it wasn't only losing to the blue, it was losing everywhere. Now its polymerization speed has been reduced and the green has recovered. So there's like a seesaw oscillation here in the activity until there is a final resolution. That's a little bit of an analysis of that, but it doesn't really matter so much. And you can see the same kind of oscillation when, instead of letting this cell spread equally symmetric on the junction, it actually arrives at the junction, which is the more realistic way in the experiment. It arrives at the junction from one of the directions. And again, you see this oscillation. So we were very interested to see if these deterministic oscillations, these are oscillations that come in the model without any noise, are actually there also in the experiment. And the idea is, yes, first of all, you see that we can have in the simulations also noise, but it doesn't change the overall structure of these deterministic oscillations if the noise is not too large. I'll show you what happens when it is in fact too large. You can see an example from an experiment. from a glioma cell. The cell looked like at first it was going to the left, but eventually it changed to the right. And what's interesting is that you can see some oscillation in the activity, which was not very easy to detect. You can see it maybe easier here, that you see these out-of-phase oscillations between the activity in the red and the green arms, which are the two losing arms, and that's exactly what we predicted. The green and red arms have these large out-of-phase oscillations in the activity. Of course, the data is much more noisy than the simulations. There is this interesting behavior that I told you about if you have actually super high activity of the cells, and that actually we saw that only for the glioma cells, for the cancer cells, which you expect to be the more active cells. And here you see that indeed the model predicts that you can get stuck in this state where the two new arms are super long because they're polymerizing so strongly. The back is completely shrunk to zero. You see it here. The back is completely shrunk to zero. You have two new arms which are competing. They're almost at the same length, stuck like that for a long time until there is a small oscillation which breaks this asymmetry and only one of them wins. In the experiment, we saw many such events. Again, you see that two of them are competing until it's resolved, but it's actually resolved in the experiment in a funnier way, because what the cell is doing is that actually it's detaching from here, which we cannot allow in the model, of course, and it's spanning like a bridge over this non-adhesive region until one of them is detaching. So it's resolving this not in the way of the model, because of the model you're bound to be in the junction. Maybe this is not so interesting. Just to show you that the effect of noise is very interesting here. If the noise is very small, then you look at a typical time in the simulation for the cell to go through the junction. It has a very nice peak, and the probability is half and half that you go into either one of the two new arms. You don't go back. If you increase the noise level in the local flows of the actin polymerization, you find that now you can have interesting behavior. There is still the peak of going through the junction at the same time that was here, with again 50-50 of going to the new arms. But there's now a new population of behavior where it takes longer, but you are going back. The blue is the direction you came from. So you almost, with the majority of cases, being reflected by the junction. So you reach a junction and the junction actually reflected you. Sounds crazy, but in fact we found an example in the experiment where the cell was doing the same thing. It wants to grow two new arms, but instead of just doing that, it just reverses direction as the model predicts. If the noise is even higher, you just have a distribution of escape times. You see the scale is huge now. So when the noise is very large, you are not able to polarize very strongly, because the noise is destroying the internal polarization that you want to build in order to go through the junction. So the escape times now is an exponential distribution, which is very long. And when you come out of the junction, it's a third, and a third which way you go, because you completely forgot by that time which direction you came from. So you have a 1/3 probability of going to any direction.
In the experiment, how do you control the noise?
You don't. But here is an example in the experiment where a cell came to a junction, and you could see that for 200 minutes was not able to make a decision on how to go through this junction. And if you look at the simulation from a typical place here with high noise, it looks very similar to these continuous oscillations. And now this is again something counterintuitive. You increase the noise and you get more trapped. Usually in physics, when you increase the noise, you untrap particles. But the problem here is that your particle, which is the cell, it has to order itself in order to move. And the noise is destroying this order. So that's why this trapping actually happens with higher noise. But at first, it's a little bit counterintuitive. I'm increasing noise and I'm getting myself trapped. That's like the opposite of what I expect. Okay, now networks. I have a few minutes to talk about networks. The experiments come, by the way, for this work from Pablo Saiz in Hamburg. Now here, you have a macrophage cell on an hexagonal network whose dimension was shrunk so that the cell is highly bifurcated at any time, meaning that it does not encounter just one junction, like I showed you before, but it encounters many junctions simultaneously. And this movie was supposed to show you that some immune cells, which are these yellow particles, when they scout for pathogens in your body constantly, in your intact tissues, they have to bifurcate in order to squeeze themselves into the cracks between the cells and the tissues, and also to sample many directions to see where, to sniff out pathogens. So we essentially, that's another example to give you motivation. Macrophages, which are immune cells, they are observed here within the tail of this zebrafish again, and you see the highly bifurcated shape of these cells. So they span many junctions. That's another example from another kind of cell that exists in your skin. So essentially what we did is extend the model that we had before. But now, instead of the flows bifurcating and splitting in two at a single junction, they continue to split more and more as they go through more and more junctions. So the contribution from the flow of this guy was split into here, but then this contribution continued here and then split furthermore there. Just to say that we don't fully believe, of course, that actin flow continues unhindered in the cell over long distances. So you can think of it more as a simplest way to represent the fact that by having polymerization here, you induce some force, some effect to polarize the cell. It's not that we think that really there are these conveyables that continue unhindered throughout the cell, but there is some effect that they induce which can be carried further down. That's the simplest thing we can think of in the way of modeling. Yeah, so let me go off that. Okay, now what we can think about, for example, is networks of different dimensions. So this is the size of the network of the hexagonal side, and this is beta, which is how active is the polymerization. So we can start here with a large network, and such that the D is large, and if D is large compared to the size of our cell, we can at most cover 2 junctions at a time, and you can see this is the number of junctions over this time series, and it switches from 1 to 2, 1 to 2, and that's the total cell length. Okay, if we go to a denser network and also increase the activity, now we can have the cell from time to time even reaching 4 junctions, like you see here. It can even cover 4 junctions, and then it can cover 3 junctions. It can go back to 2, etc. Just to give you a sense of what is it doing. When we compare just the effect of the size of the network, you can see that when the network is smaller, then you can reach a higher number of junctions compared to when the network is larger. So we're comparing this point with this point. How does that affect diffusion? So the denser is the network, so that you reach larger number of junctions, for example 3, 2, and 1 instead of just 2 and 1, it means that you get more arms, because you cover more junctions, you have more arms. If you have more arms, you have more competitors for this polarity, and that means that indeed the average maximal difference of this polarity cube between any edge points along your cell is on average smaller than if you have less arms. So the more arms you have, you are less polar. It's more difficult for you to polarize. You get more easily trapped. It's more difficult for you to go on moving. You're less persistent. And you can see that in the effect on the diffusion coefficient. So if you actually now look at the mean square displacement of these particles on these hexagonal networks, first of all, it is looking like normal diffusion, at least under these conditions. But again, this is for low noise. What happens at high noise, and we know that when the noise will be large enough, you'll get trapped. So you won't be moving. So is there something interesting between the normal diffusion and the trapping? I don't know. That could be an interesting thing to think about in the future, to explore. if you look at the diffusion coefficient, and of course, trivially, it increases as you increase the activity of the polymerization, so the cells are moving faster. That's easy to understand. So why it's getting darker to the right, that's easy. And denser networks, which are lower here, they get more bifurcated cells, so less persistent and lower diffusion coefficient. So if you make the network size larger, you're diffusing more, because a cell cannot have many arms because the cell length is the same. And now the cell can actually diffuse faster. It's more persistent. You make the network more dense, the cell has more arms, it can diffuse much less efficiently. You can even see that, I think, with your eyes here, because the less dense network is here, and this guy actually gets further from the beginning than this one, which is the one in the denser. And the difference is not very large, but the diffusion coefficient is different, and you can even see that with your eyes. That's how the trajectories look here. Of course, look like here. Of course, you can see the traces of the hexagonal network underneath. But as I said, it's still simple diffusion, not anomalous at this stage. Yeah, let me just show you what does the noise do. So if you look at load noise, which is the movie that I showed you before, then you can now ask, what is actually the residence time of the cell on configurations of 1 junction, 2 junction, or 3 junctions. And what you can see, which is interesting, is that there is a special high residency time on shapes with 2 junctions for this particular choice of size of network, and they dominate. Now, if you increase the noise, what happens is that, if you increase the noise, what happens is that the residency time increases, and we know that the residency time, the mean residency time increases because of what I showed you before. Remember that when the noise increased, you can even get complete trapping on a single junction. So the same kind of behavior here. So lower diffusion coefficient when you have larger noise. Let me jump over that. Just we can compare a little bit, mostly with the eye, the experimental data with simulations, and we can even extract the residency time distribution on junctions in the experiment and compare it to the simulations. For this particular configuration, the difference between covering 1 junction like here or 2 junctions like here has similar residency time distribution. If you look at the movies, you can see this is the UVEC, that's the endothelial cells. These are not cancer cells. They are not very motile, at least in this example, and in the corresponding simulation. You can see this one gets stuck here for quite a while before it starts moving away. But there's examples of the same kind of cell, but just more motile. So there is always a distribution of behaviors, even in the normal population of cells. So again, you see this one is much more motile. And again, we had to crank up this activity parameter from 7, which was in the movie I showed you before, to 9 in order to simulate how this cell has its behavior. So clearly, this activity inside the cell is variable between cells in the natural population. Yeah, again, this is the macrophage, which is naturally a much more motile cell. And again, we can compare the simulation with the model. So I'll just summarize here. We started with a simple 1D model for how cells can have various migration patterns. We extended it to looking at how a cell deals with a single junction. We found this interesting deterministic oscillation before it makes up its mind, as if it makes up its mind, and interesting effects of noise. Then we extended it to this highly bifurcated cells which move on a network. And that's now on the archive. Open questions. I think many open questions, but you can please come up with others. For example, biologists are interested in what is the optimal shape for these immune cells which are scouting your body. You want to have many arms maybe, because if you have many arms, you can sniff out very well a pathogen. But if you have many arms, you are not really able to move very efficiently. Maybe you can solve it by arms formation in transient bursts. You make lots of arms to sniff, and then you shrink down the arms in order to make much more efficient migration in the direction that you sniffed something. I don't know what are the biological solutions for this sort of optimality question with regard to the size of this cell, because the network that these cells have to span, that's given by the tissue. How does chemotaxis or response to other gradients affect migration in such networks? What happens if you are in the dense system and instead of moving alone, you're moving in pairs? Like you see this pair, for example, is really moving together like a small train. How do you behave then? Is it maybe more efficient to move like that on such a network? What happens when the network is not a pure hexagonal network? Does that actually make a very big difference? What happens if it's a completely random geometry or a random network where you just make random networks? We know how to produce them. We haven't checked yet what the model would give. That's an example for a macrophage migrating in the zebrafish, and clearly you can recognize junctions, but they are clearly not on a purely hexagonal network. It's a hopping type of random motion, right? Because you can be almost not motile, like this one, which is trapped, and you can be trapped transiently, and then you can actually begin to move, or you can move almost smoothly like this one. So as function of size, noise, and the intrinsic activity, can we find anomalous diffusion within this model in some regime? I don't know. But since it's of interest to this crowd, I'm raising this question, but I'm also interested in it myself. And thank you.
Thanks very much. Other questions, comments?
Two questions, both related to anomalous diffusion. So first, in view of your detailed model that you constructed at the beginning based on the actin flows and so on, is there any mechanism to generate anomalous diffusion from it, or does it only generate normal diffusion? And secondly, exactly on that, it may be interesting to look at the waiting time distribution. If it could be that the transition to localization could be marked by a subdiffusion. If so, you should see that in the waiting time distribution crossing over from exponential to power law.
Yeah. So to your first question, in the 1D model that we started with, we have a regime. Well, we studied it extensively for the fixed length model, which is the simpler model that we started with. And for that model, we don't have stick slip and all of that, but we still have different regimes of random motion. And we have, in particular, random walk, we have persistent random walk, and we have intermittent random walk. So the intermittent random walk can look like run and tumble under certain conditions. And we thought that maybe it can give us some levy flights and things like that. It seemed not to. Here, we haven't studied it as extensively as of that previous model with a fixed length, because once the length gets variable, it's more complicated to clearly characterize what is the type of motion that you have here. And as you saw, all the waiting time or residency times distributions that we have found so far are exponential. So it's actually a good question of what needs to be added to a model like that to make it anomalous, to change the exponential waiting time distribution to something which is more like a power. What are we missing if we want to go into that regime? Or maybe it's still somehow hiding within this model, which is not so simple. Still, it's not so difficult compared to how a cell really works, but it's not trivial. And maybe we somehow missing something in there, but we haven't found any evidence of that so far. Yeah. It's a good question. What is missing? What should be added to make these guys have a power law distribution of residency on a junction? Yeah.
So I have a question. I mean, if you as you increase the strength of the noise, the sigma, so you see that of course it has a higher probability to get trapped. Is there a sort of sharp critical value of sigma c beyond which it gets trapped, or
so it doesn't look like that, because if you look back, I mean, where we studied it most clearly, which was for the single junction, you see that in the limit of very high noise, we just get this. We are slowly transitioning from a distribution time. Here, you were slowly transitioning from a distribution of times to escape, time to go through the junction, which is multi-peaked, to something which is just a pure exponential. So how do you say when have you shifted from something like that to a smooth distribution? It's not very clear. Yeah.
are there?
Yes. Maybe my question is related to that. I'm interested in how you would model the state of this little thing. What is the state variable you think of that you're allowing to evolve? What are the state variables that you need to keep track of to understand this behavior?
You mean of this of these guys?
Yeah.
With the many arms.
Yeah.
Well, the simplest way we have so far characterized such an object is by its number of junctions and arms.
That's enough.
Because it's you know, this shape is made out of these linear segments, but it's not enough, because you can have the same shape, but the internal flow pattern can be different. So in fact, it is already highly dimensional, high dimension for just describing exactly the instantaneous state of these objects, because you have really to give me what's the instantaneous flows on each of these tips, and of course then the number of these tips and the number of junctions. Then I know everything about this object at each time. Yeah.
Can you then learn behavior?
I'm sure you can. We haven't tried that. Yeah, like to cluster these behaviors. Yeah, that would be interesting.
Other questions, comments? Okay, if not, let's thank